Good afternoon, distinguished participants. First, I would like to welcome you all to this forum. It's my pleasure to represent Dr. Wang to host this forum entitled Research Progress in Depressive Disorder. Before we start this forum, I would like to give a brief introduction about depression. Depression is a common mental disorder. About 5% of adults suffer from depression worldwide, and the onset age of depression becomes younger and younger. The prevalence rate of women is much higher than that of men. The efficacy of antidepressant is greatly different among individuals. In recent years, some important research achievements have been made in depression, and the new mechanisms and potential targets for depression have been found, which provides a new idea for the research and the development of new antidepressants. In this forum, we invite two experts and two young scholars to elaborate relevant research progress and the achievements from the aspects of epidemiology, psychopharmacology, proteomics, and neuro-imaging. Okay, now let's welcome the second speaker. Okay, first I will introduce our second speaker. The second speaker is Professor Jing Du. She is also from Beijing Anding Hospital, Capital Medical University. Professor Du is a former senior researcher of the National Institute of Mental Health of the United States. She is a recipient of Beijing Phonics Talent Program. She has participated in a major project of brain science and brain-like research. Now she commits to the research and the development of antidepressant and anti-autism monomer innovative drugs. Next, she will introduce her research about development of fast-acting antidepressants based on nuclear receptor mechanism. Now let's welcome
Thanks for the introduction. And I'd like to thank the organizer to give me the opportunity to share my research here. Today, I'm going to talk with you the drug development, the novel antidepressants we developed by the nuclear receptor mechanism. Okay, as you all know, there are about 300 million people in the world has major depressive disorder, and about a third of people do not respond to the the current drugs, and so there is an urgent need for development of drugs with novel mechanism. And our group is interested in developing novel antidepressants from the single molecules isolated from the herbs. As you all know, that the depression is associated with many factors. It can be environmental, it can be epigenetic events, or genetic factors, the susceptible genes, or the endocrinology, hormonal regulation. And so currently the cellular and the molecular pathophysiology is still unknown, but people knows more and more about it, and they're trying to put the pieces into a picture. The neuro-immune balance as a mechanism to develop antidepressant drugs is, cause a lot of attention. because the meta-analysis shows that shows very consistent clinical findings, and they found that the CRP levels, TNF-alpha, or interleukin levels are altered in the depression patients. And those factors also associated with the drug-resistant depression and even people with the suicidal ideation. Then people developed a new mechanism, the neuro-immune mechanism for depression, where they think that depression is caused by all this danger-associated molecular patterns or the microbial-associated molecular patterns, and they activate the inflammations and cause the increase of IL-1 or IL-18, and which leads to more inflammation. And also, caspase-1 could in turn to inhibit the excitatory Ampa receptor activity that also contributes to the depression behavior. In addition, the mitochondrial function also contribute this. And you can see that in people with depression, the mitochondrial DNA is damaged, and the ROS is also increased. The membrane becomes more permeable, etc. So we call this depression mitochondria in comparison with the normal ones. With all this change, that the mitochondria, the damaged mitochondria can activate the formation of inflammation again to activate the inflammation, you know, the release of IL-1 beta, IL-18, etc. So that's how mitochondrial function contributes to MDD. Dr. Ron Duman has a very important review in molecular psychiatry that he has summarized all the factors that affect the fast antidepressant effect of ketamine, that he thinks the enhance of GLOR1 at synapses, the enhanced BDNF expression, and modulation of autophagy all are important for this process. So now here, why nuclear receptors? Nuclear receptors such as glucocorticoid receptor, GR, mineralocorticoid receptor, MR, and the estrogen receptor, ER, like Dr. Rana Lee just said, you know, sex matters, and those sex-related receptors, and also aryl hydrocarbon receptor, AHR, and the PPA gammas, etc. Those nuclear receptors are quite powerful. They can, after activation, then can activate the transcriptional factors, and then also modulate the inflammation activity, so that those nuclear receptors are that we want to study nuclear receptors to find new medications. It is also important to study from the endocrine perspective, that we found the agonist of those receptors. To find the agonist of those receptors all are important for develop novel antidepressants. So here we gave examples that how nuclear receptor matters. We all are interested in ketamine, which really has an antidepressant effect. And this study started from John Crystal. John Crystal found that this ketamine have a very fast antidepressant effect in patients. Then Carlos Zarate and his colleagues replicated this finding and also extended this finding and found that low-dose ketamine can get an antidepressant efficacy within 2 hours and last for a pretty long time. Other groups, and including our Dr. Gang Wang's group in Anding Hospital, further tested the effect of ketamine and bipolar depression and the suicidal ideation, and find that this drug is quite effective. And now Dr. Husseini Manji's group in Janssen Pharmacology, the Janssen Farms has developed the nasal spray for the treatment of depression. And all this are pretty good achievement during the past 10 years. And in terms of a mechanism, that people find that ketamine is could inhibit the pro-inflammatory cytokines like IL-6, TNF-alpha, etc. And also after about 1 or 2 hours, the quinolinic acid and quinorenin, those molecules are increased in the plasma of the patients. And as a consequence, also that you can see Ampa receptors, BDNF, mTORs, implicating that the autophagy process all are activated. To give a very close look to this mechanism, that you can see the tryptophan metabolic pathways, and usually tryptophan can change into 5-HT, but and the inflammation process that it can turn to quinorenin. And then during chronic inflammation process, the quinorenin can change to quinolinic acid, with the help of KAT, another enzyme. Those two molecules both can activate AHR. AHR is an inhibitor for inflammation, and within here you can see that with the activation of AHR by quinorenin and quinolinic acid, and the assembly of inflammation is inhibited, and also the production of caspase-1 is inhibited with a lower level of IL-6 and IL-1, and the inflammation are inhibited. So this is currently, people think even ketamine go through the nuclear receptor mechanism to achieve its effect. The other nuclear receptor, which all of us are familiar, are the glucocorticoid receptor. When I just started my career 20 years ago, and Dr. Manji mentioned the importance of the cortisol, because in Cushing's syndrome, that people has hyper cortisolemia, and the level of cortisol in the blood is pretty high. When you get the surgery to remove the tumor, and the mood was immediately improved. So that just gave us implication that the nuclear receptor GR may play a very important role in the development of depression. And the most consistent biological change in MDD patients are the elevation of plasma cortisol and overall the HPA axis. A recent meta-analysis also find that HPA axis and depression shows a very close correlation, and the people are drug resistant are usually with chronic elevated cortisol levels. Also that, in the clinic with patients with child trauma or long-term stress, their response to the medicine are not so good. Why? Why the nuclear receptor is important in this situation? So in MH, I've been working on these receptors, and we've published a couple of papers on the glucocorticoids. And what we found is that the glucocorticoid has two phases. One is the acute phase, and during this phase, you can see enhanced mitochondrial function, inhibition of inflammation, and enhance the Ampa receptor excitatory synaptic function. While after long-term chronic massive glucocorticoid elevation, and this effect is reversed. And we measured all these chaperone proteins of GR, like FKBP51, like BAG1, all those receptors, all these chaperone proteins are reduced. I mean, they just suggest that they're exhausted. So in this situation, the activation of glucocorticoid will have the opposite effect, that you can see that the decreased mitochondrial function, the inflammation is activated, and also decreased excitatory synaptic transmission. This is how GR works so far, we think. Another example I can give you for the nuclear receptor is vitamin D receptors. You know that vitamin D deficiency may also be the cause of the drug-resistant depression. Vitamin D deficiency is pretty worldwide, is pretty common that the children or adult worldwide, that many people has vitamin D depression. And there is a correlation between low vitamin D levels and depression, and a patient with vitamin D deficiency will have more severe depression symptoms. And recently also meta-analysis showed that vitamin D treatment supplementation is clinically effective for the patient with depression. And this study may be useful for clinical doctors to see that, their vitamin, the patient, if they're drug resistant, if the vitamin D level is abnormal. What is the reason that vitamin D regulates the depression? And one vitamin D receptor, VDR, they are nuclear receptors. What they do is they inhibits the inflammation like TNF-alpha, IL-6, IL-1 beta, etc. And also helps with the mitochondrial function. So, with the deficiency that you can imagine that this antidepressant, anti-inflammatory effect is not there anymore. And the VDR is possibly went through the anti-inflammation to exerts antidepressant effect. The sex matters, the sex hormone matters. Those are, testosterone and also estradiol. So those sex hormones, they can go through the ER and ER receptor, estrogen receptor. Estrogen receptor could modulate the mitochondrial function and also has an anti-inflammatory effect. I will share you with a review that, pre-menopause normal areas, normal levels with circulation estrogen that you can see with this, the estrogen will protect mitochondria. The estrogen receptor beta is responsible for that, and also inhibit the assembly of inflammation. During the perimenopause, where the estrogen is insufficient, and you can see that there will be assembly of inflammation and the production of interleukin-1 beta, etc., to have like an inflammation activity going on. Therefore, the levels of estrogen is quite important, and the perimenopause depression is quite popular. To make a summary of the research I've been telling you on those activators like ketamine, like cortisol, estrogen, vitamin D, they all work on the nuclear receptors like AHR, GR, ER, VDR. And to exert its anti-inflammation formations function or anti-inflammation function that will inhibit the formation of caspase-1 and in turns to keep the Ampa receptor activating to inhibit the pyroptosis and also inhibit the release of IL-1 beta and IL-18. And those effects may be beneficial to the depressive behavior. And so are interested in our monomers. And we want to look at the herb monomers that is effective. During our research, we found that some Chinese herb monomers are metabolized of the vitamin of the bacteria in gut that a potential pathway for the microbiota gut-brain axis communication, maybe those metabolites. And in the microbial derived metabolites, there are two systems. One is the tryptophan metabolism that I've mentioned to you. This process also mediated in the intestine and the gut in the gut, that it can develop a lot of derivatives which affect eventually to the brain. And the other system is the bile acid metabolism that, the bile acid can be a very important modulator for the brain function as well. We have a close look to this pathway, and you can see that in the liver, the primary bile acid were formed from the cholesterol. They were synthesized like CDCA, like CA, and those are conjugated by taurine or glycine, and the bacteria is responsible for the deconjugation of those bile acids. And so those bile acids can get into the blood and pass blood-brain barrier, and also affect the function of the brain. About 70% of the bile acid in the brain are synthesized by the liver or intestine, and about 20 to 30% are synthesized in the brain locally. So to test if this hypothesis is right, we want to see if the CDCA, does CDCA have a fast antidepressant effect or not? So what we does is we IP injected the CDCA into the C57 mice. Then after 60 minutes, we look at the tail suspension test. 3 days, we'll look at the open field test, and 5 days, we'll look at the forced swim test. Those are tests we regularly to test the despair of the animals. And we found that after 60 minutes of treatment, there is a significant antidepressant effect by CDCA, which is an endogenous molecule that is quite important for our mood. And after 5 days of treatment, and you so can see there is a antidepressant effect, because we know the receptor for CDCA is FXR, that we want to see if the FXR specific inhibitor Csds can inhibit the antidepressant effect of CDCA. And you can see that with the CDCA, you can see an antidepressant effect compared to the control. But with the application of Csds, then the yellow bar, you can see that the antidepressant effect of CDCA is gone with the inhibition. This is a green bar, that the antidepressant effect of CDCA is gone. So it probably is mediated through its nuclear receptor FXR. We also look at the open field test, the travel distance in the center area, and also the total travel distance. It seems like those animals after treatment is not hyperactive, and the weight also show not much change. Then we look at this effect in the Csds animal models. So the experimental procedure is we established 10 days of the Csds procedure, and then we use the sucrose consumption to and also tail suspension to look at the effect of depression if our animal is successfully establish the depression model. And you can see that the preference, the sucrose preference in the after CSDS treatment, after CSDS procedure is lower. That means it's a little bit depressive. And the immobility time is increased in CSDS. But the CDCA treatment can rescue this depression effect from CSDS. And after 5 days, it also shows inhibition of this depression effect. So that indeed a CDCA in this animal model of CSDS also show the antidepressant effect. And what is the mechanism? Does it, previous study have shown that activation of FXR can inhibit the inflammation activity. So we found that in this CSDS animals, just the FXR level are decreased significantly, and after treatment with CDCA, and we restored the FXR level. And after CSDS procedure, the depressed animal showed increased pro-caspase-1, caspase-1, pro-IL-1 beta and IL-1 beta, but treatment with CDCA significantly inhibited this effect. Then we'll look at also the Ampa receptor expression level. After the CSDS procedure, the depressed animals showed decreased expression of phosphogluR1 and GluR1. Phospho-A45 is decreased. After CSDS treatment for 5 days, and you can see that the Ampa receptor expression is increased. Make a summary of the first part research that the CSDS decreased the FXR expression level. Well, after CDCA treatment for 5 days, the FXR level was restored. Also, CDCA also inhibited the NLRP3 inflammation activity and caspase-1 activity, and also increased the level of phospho and expression, phosphorylation and the expression of GluR1. CDCA showed that antidepressant effect in test of sucrose consumption, tail suspension, and forced swim in CSDS mouse model as well. So here is the schematic conclusion that liver and intestine synthesized the CDCA, and it passed blood-brain barrier and get into the brain. In the brain, it can inhibit the inflammation activity to reduce the caspase-1 and also enhance synaptic Ampa. This may be the mechanism for the CDCA. So the second part of research, we want to look at the bile acid receptor FXR. Can that be a target for the development of new drugs? And then we found like a ganoduric acid A. We do a molecular docking and find ganoduric acid A can fall into that pocket. Ganoduric acid A is isolated from Ganoderma lucidum. that in China is quite famous. Lingzhi is a very famous herbal medicine, and in Asian countries. Ganaduric acid A, GAA, is isolated from this mushroom, and it is very, I mean, it's about 80% of its effective component. It has an anti-tumor, antibacterial, anti-inflammatory, or immunosuppressive effects. Flavonoid X receptor is a bile acid sensor and receptor. It also, it's a receptor for CDCA. It plays an important role in regulation of lipid metabolism, but in terms of mood regulation, that we probably is the first to investigate that. So our first question is, does the GAA have a fast antidepressant effect in first swim test or tail suspension, or the CSDS, the chronic social defeat stress animal models? So this is the structure of GAA. And what we've done is we treat the animals with a GAA, and after 60 minutes, we did the tail suspension for swim, and also after 5 days, we did both. So here's the result that after GAA in various concentration treatment, which is an IP injection, and you can see that there is a pretty fast antidepressant effect. After 60 minutes of GAA injection, and you also can see in first swim test, there is an antidepressant effect with a reduction of immobility times. After 5 days of injection, and the effect remains in tail suspension and first swim test. And the open field test didn't reveal any hyperactivity. So the animal is still pretty normal, and the body weight doesn't change much. And the distance travel in the center, you can see a slight insignificant increase. We also look at the drug effect in the CSDS animal models, and you can see that we spent 10 days to establish this social defeat depression model, chronic social defeat animal depression model, that we, at the 13 days, we did the sucrose preference test and the first swim test. And then we choose the one which is depressed, the animals which are depressed, and then apply GAA for 5 days. And then we look at the first swim test and look at the antidepressant effect. If GAA 2.5 milligram per kg does show antidepressant effect in the CSDS animal model. So our next question is, can GAA inhibit inflammation and activate the Ampa? So with the animals that we, after the behavior study, we isolated the prefrontal cortex, and then did a Western blot analysis to look at the NLRP3 levels, the inflammation levels, the pro-caspase-1 levels, caspase-1 levels, and the pro-L1 beta and the L1 beta levels. You can see from the examples represent the actual gels. We did a statistical analysis, and you can see that after CSDS, all those factors are increased. And after 5 days treatment with GAA, and this increase were rescued by the treatment of GAA. And then we look at the Ampa receptor levels that you can see after CSDS procedure, you can see decreased Ampa phosphorylation and Ampa also receptor expression of GLUR1 and GLUR2. After 5 days of GAA treatment, and this Ampa receptor phosphorylation was increased, and also GLUR1 expression level was increased, so does GLUR2. So the third question is whether GAA have this antidepressant effect through FXR. So first of all, we did a molecule docking that we found that, for the FSR and GAA, you can see that it can really fall into that pocket, and also a couple of residues, or they have like a hydrogen interactions. And after treatment with various concentration, the FXR levels, this is our FXR cell numbers and expression levels. Those are all increased after expression. After GA treatment, there is an increase in FXR expression. In the CSDS animals, you can see there is a significant reduction of FXR, but it's rescued by the CSDS, by GA treatment. So that the FXR, the anti-inflammatory effect, can be rescued. We also used FSR knockout mice, and we found that this knockout mice shows depression-like behavior with very low level of FXR, and the animal showed depressive. And that with the GAA treatment, there is a slightly increase that this is reduced. The antidepressant effect of GAA is reduced. And this is the FXR levels, and there is still remaining a little bit of FSR that so in FXR knockout mice, the inhibition of the inflammations are abolished. That you can see that in normal controls, you can still see a GAA effect on the pro-caspase-1 and caspase-1 levels, but in the PFC of the knockout mice, you cannot see the anti-inflammation activity. Make a summary of the GAA story. I've told you that ganoduric acid A showed a rapid antidepressant effect in chronic social defeat stress depression model. Tells us tail suspension test and forced swim test. And the GAA directly inhibited the activity of inflammation and activated the phosphorylation and expression of Ampa receptors. FXR expression is decreased in the Csds depression mouse model, suggesting that FXR plays an important role in the pathophysiology of depression. Meanwhile, the FXR gene knockout mice showed depression-like behavior. In FXR knockout mice, or using FXR-specific inhibitor, Csds, the antidepressant effect of GAA and also the inhibition effect of inflammation what were abolished. The third story I would like to tell you is another nuclear receptor, pipa gamma. We use the oridonan, which is the bioactive component of Rhabdosia rubescens. It's also a traditional Chinese herb. And oridonan demonstrated an anti-inflammatory effect in various disease models. And pipa gamma, it has been shown to be associated with depression-like behavior by many studies. And but whether or not oridonan went through pipa gamma or Ampa receptors to exert its antidepressant effect is still unknown. So what we've done is we, after 7 to 8 weeks CD1 mice, we IP injected oridonan into those mice. And we inject it after 1 hour or 5 days. And then we look at the different tail suspension, first swim, open field test. And we found that after 1 hour of injection, and there is a significant antidepressant effect. And also in first swim test, we found the same thing. Is this antidepressant effect sustained through 5 days, and after 5 days of injection, you still can see in tail suspension test or first swim test, there is a significant dose-dependent antidepressant effect. To look at if it has a hyperactive activity, and we also subject those treated animal in open field test, and it seems like they are not hyperactive. Next we look at the expression levels of pipa gamma, and after treatment with oridonan, and there is a significant increase of pipa gamma level. And also we use this GW, which is a pipa gamma specific inhibitor. Then when we apply it, the oridonan effect, antidepressant effect is gone. And next, we also look at the Ampa receptors, and we look at the Ampa receptor after treatment with 1 hour or with the after 5 days. And also you can see that Ampa receptor activity is increased. The phosphorylation is increased, but GLUR2 or GLUR3 do not show a very significant effect. We also prepared the synapsomal preparation to look at this receptor levels at the synapses. And this is the enrichment of this preparation with the synaptic proteins. And in the synapse, we found pretty similar that the phosphorylation in the synapse of GLUR1 and GLUR, phosphorylation and expression of GLUR1 is increased. Well, the GLUA2 after 5 days and GLUA3 after 5 days also show the increase. Then we'll use the GAIKI, which is an Ampa receptor specific antagonist, and 30 minutes before we do the oridonal injection, we use the GAIKI, and then we did a tail suspension test and found that the Ampa receptor antagonist GAIKI can abolish the oridonal effect, antidepressant effect. Make a summary of the research that after treatment with oridonal, the immobility time of mice was significantly reduced in the tail suspension and force film test. And also after treatment, the pipa gamma was able to, the expression level of pipa gamma was increased, and the blocking pipa gamma was able to block the antidepressant effect of oridonal. Ampa receptor GLUA1 phosphorylation and GLUA1 expression was increased both in the synapsomal preparation and the protein extract from the prefrontal cortex. And the antidepressant action of oridonal can be blocked by Ampa receptor specific antagonist GAIKI. So make a summary that based on the nuclear receptor regulation, and we are trying to develop novel antidepressant. And those antidepressant like GDCA, which is an endogenous agent, and GAA and oridonal, they are both from the Chinese herbs. And they seems like mediated through nuclear receptors, they can inhibit the inflammation activity, and then subsequently activates Ampa and benefits depression-like behavior. So in our experience, the target as a nuclear receptor are really beneficial, and I think those directions is warranted for clinical studies. Thank you. And I would like to thank the Beijing Anding Hospital for all the support, and also this work has been done by some my students, Bao Hongkun and Li Haoran and Liu Ping in Yunnan Kunming University. And I would like to also to thank the funding and from the Beijing Hospital, Beijing Anding Hospital, and also the nation's NSF funding program. And thank you.
Thank you for Professor Du's presentation. She just introduced her team's funding, that some components and chemicals which can activate the nuclear receptor FXR can inhibit brain inflammatory activity and play a rapid antidepressant role in chronic stress-related depression. This can be the new target for treating depression. Thank you for your presentation. I think there's a question for you.
Right.
Okay, the question. Okay, only one question. Does other bile acid, okay, 其他的胆汁酸 show similar antidepressant effects?
Right. Thank you for the question. I think it's a very important question. And for the CA, they also activates FXR. So we think probably CA has a similar effect. As to others, we haven't tested yet. These are like unpublished data. So far we just test CDCA. But both CDCA and CA, they are agonists of FXR. So I think that probably both this two possibly have an effect. But the other like LCA or others, we don't know yet because they're not very good agonists for FXR. In terms of affinity, CDCA has a higher affinity in comparison to CA. So possibly the efficacy of CDCA could be higher than CA. This is a very good question. We're working on it, and I think maybe I'll report next time. Thanks.
Okay, thank you. Okay, next, let's welcome our third speaker, Professor Zhi Yang. She's a young scholar. Professor Yang is from Shanghai Jiao Tong University. He is a principal investigator at Shanghai Mental Health Center and Institute for Psychological and Behavioral Science. He works on developing novel neuro-imaging methodology for understanding individual differences in brain development and mental disorders. His team developed a naturalistic imaging into an individualized inference method based on normative models of brain responses. He will demonstrate the application of this method in his speech entitled Naturalistic Imaging and Normative Models for Individualized Inference in Mental Illness. Okay, the time is for Professor Yang.
Okay, can you hear me well? Hello, can you hear me?
No problem.
Okay. Hi, everyone. I'm Zhi Yang. Thank you for the introduction, Dr. Xiao. It's my great pleasure to present my works on this forum. I want to share some of our efforts on developing novel imaging paradigm towards individual inference of brain functions in mental disorders. So when we talk about brain functions, we are actually dealing with a spectrum of brain functions from very basic elements to highly integrated systems. So I have listed here some psychological terms that correspond to different degrees of functional integration. Corresponding to this functional spectrum, we also have a series of approaches to measure the brain functions across the spectrum. So biologists and psychologists have developed various methods, for example, to measure elementary brain functions, such as the sensory, attention, and perception. Psychophysics approaches has been developed. And for measuring brain functions related to language, metacognition, memory, executive functions, etc., repetitive cognitive tasks have been developed. But for much more integrated brain functions, such as the personality and mental health problems, the current measurement methods are mainly questionnaires, interviews, and observations. So on the one hand, based on the idea of reductionism, researchers have linked the problems in the most integrated functions to the many elementary functions. For example, studies have reported attention bias in affective disorders, such as major depressive disorder. However, on the other hand, the integrated functions may not be simply a combination of the elementary functions, but the quantitative and objective measures for these integrated brain functions are still rare. So we need more quantitative and objective measures for this problem. Now, let's look at the difficulties we often encounter in studying the neuro-imaging markers of the mental illness. The nature of the highly integrated brain abnormality determined that various psychiatric symptoms reflect a wide. This fact leads to high intersubject heterogeneity in brain functions. This issue has been one of mental disorders because we have difficulties to draw a general conclusion for a given group of patients. So on the left half of this picture, we can see that we often assume when comparing the groups, we often assume the two groups have heterogeneity for themselves. For example, the red hats in this picture represent a reference group, such as healthy control subjects, and the blue hats represent a group of patients. So we assume that these two types of groups have high homogeneity within each group. In other words, the subjects have higher similarity within the group than between the groups. No matter what we use, what method we use, we may use classical statistical tests or some sophisticated machine learning algorithms. Our goal is to find a boundary between these two groups of samples. And if a clear boundary is found, it is called a neuro-imaging marker that distinguishes the two groups of people. However, because we do not clearly understand the relationship between mental symptoms and the abnormal, highly integrated brain function, the groups we divided according to mental symptoms may not have intra-group consistency in brain function abnormalities. This situation is shown in the right half on the right half of the picture. So you can see that although the blue hat, assigned into a group based on certain symptoms, they may not be similar in terms of abnormal brain functions. This situation will cause the heterogeneity of our research samples, which will affect the statistical power of the research and the stability of our conclusions. Such unstable research conclusions are not uncommon in imaging and genomic research. So one possible strategy to solve this heterogeneity problem is the normative model. This model is very conventional. It's a very conventional strategy in the measurement of psychological properties, such as intelligence. So when we compare each person's performance with a fixed norm to get individualized inferences, we call it a normative model. In this process, we do not need to assume a certain group being tested has the same characteristics. For example, we do not assume the similarity of intelligence across the people we test. So the paper I'm showing here was published in 2018. This paper demonstrates the application of normative methods in brain imaging of mental illness. At the same time, this result also support the theory we just saw. It says among the patients with the same diagnosis type, this study only finds that only 2% of the patients had the same abnormal brain structure. This heterogeneity within the group is so great so that we can hardly expect that comparing this group of patients with healthy controls can obtain stable brain imaging results. This observation supports our use of multivariate normative model to infer the patient's brain abnormalities. So to provide a very brief summary of the background, we need neuro-imaging paradigms that simultaneously possess two features. First, they review brain abnormality in integrated functions in a quantitative and objective way. Second, it allows individual inferences so that our conclusion are not only stayed on the group level. Next, I'm going to introduce our ideas and works of developing naturalistic imaging into an individualized inference method based on normative models. I'm going to present 3 studies. The first one demonstrates the principle and the features of the method. The second one provides empirical evidence for the reliability of the approach. And the third presents an application to assess the maturity of social emotional functions for children and adolescents. Let's start by introducing the naturalistic imaging. So functional imaging reflects the working state of the brain. However, due to the state dependence of brain functional measure, functional imaging usually suffers from the influence of various noise, confounding factors, and the difficulty of data acquisition. Naturalistic imaging provides a way for alleviating these problems. In 2004, Hassan and colleagues published a paper on Science showing that watching the same movie can induce synchronized brain activity among different persons. So many subsequent analyses have shown that this synchronized brain activity between individuals reflect similar understandings of the stimuli. Natural stimuli such as movies, music, and dialogues provide rich interactive contents that are closer to the reality, making the naturalistic imaging a promising and objective way to reflect the integrated functions of the brain. In addition, some studies have proved that compared with the traditional resting state and the repetitive cognitive tasks, naturalistic imaging can enhance the engagement of the participants, reduce their head movement, and increase the success rate of image acquisition. This advantage is particularly important for research on mental health, children, and the aging population. So in order to give you an intuitive display, we have made two videos, both of which are derived from our research. So let's start first, the video on the left. So as you can see, the similarity of the brain activities of the two subjects are relatively low in most scenarios. And the similarity of the two subjects are indicated by the height of the bar in the middle. So in some scenarios, let's see now. Okay, now you can see in this particular event, the synchronization of the two subjects become significantly high. So this phenomenon suggests the naturalistic imaging can reveal common brain responses to realistic events and therefore help decode what is in common in our brain functions. So this is very important for the basic part of the research to let us to exclude some subject-specific factors that influence our result. Now let's start the other video. Okay, this video shows the result of similarity of a multi-wideband EEG map. So you can see the correlation matrix on the right bottom corner. So as you can see, the average actually we are showing this movie to 6 different individuals, and the correlation matrix shows that different individual similarities in different scenarios. It changes with the context of the movie. At the same time, you can see on the right upper corner of the matrix, the color shows the average similarity across the 6 individuals. So this matrix change with the movie. This phenomenon not only reflects that the synchronization of all subjects' brain activities, but also reflects that certain scenarios in the stimuli can evoke individual differences. Therefore, we have the potential to detect where and when in the brain is where and when a brain is different from the others. So, sorry. So this slide demonstrates our idea to combine the naturalistic imaging and the normative model to tackle the heterogeneity problems in the mental disorder research. So when we present natural stimuli, carefully designed natural stimuli to all subjects, we can make normative models of brain responses of the reference group. In this picture, the red hat. If we pull the responses of all the red hats together, we can get a distribution of brain responses of the reference group. The reference group represents the mean and the variance of the reference population. Then, for a given subject, we can compare the individual brain response to the distribution of the reference group. And this allows us to make individualized inference for that given person. The idea is widely used in psychological assessment. However, the difficulty here is how to optimize the effect size. In fact, we need to identify brain response patterns or features that have relatively low variance. That means we have to narrow down the variance of this distribution, and also evoke the difference between the blue hat and the red hat. In that case, we can clearly find the abnormality of the given subject. So naturalistic imaging provides a chance for searching for low variance features in the reference group, because as we seen on the last slide, it is capable to evoke synchronized brain responses. And it also helps review individual differences among the patients and the reference group. So here I'm gonna show a study we have published according to this idea. So in this study, we have established a methodological framework for implementing the idea. We verified the effectiveness of this method by identifying patients with schizophrenia. So we first customized a piece of video material. It contains plots such as arousal of various emotions, and they change they change along the time. And we also include metaphors and analogies. We expect that certain episodes of the movie can make mentally healthy subjects have similar feelings, and thus they will have similar brain activities, while the schizophrenic patients may have a different brain activity patterns from the healthy people due to different internal experiences. Please note that in this paradigm, we do not assume that all the schizophrenic patients have the same feeling or have the same brain activity patterns. So this is a difference from a traditional group comparison study. So we try to find the individual differences, and use these features to determine the state of the disease. So after obtaining the fMRI data of 62 schizophrenia patients, here we noted as SC, and 44 healthy control, here we noted as NC, normal control. When they were watching the video, we need to determine which episode will induce relatively consistent brain activities in healthy people, and where these activities are in the brain. So here we use a sliding window approach to analyze the data piece by piece, so that we will show in each context or in each clip of the movie how consistent are the people. So we calculate the ratio between the inter-subject correlation noted as ISC of healthy subjects to the patient's ISC. The ISC indicates the concordance of the brain activity among subjects, which represent the inverse of the variance. So that means higher ISC indicates lower inter-subject variance. So by comparing the ISC of healthy control and ISC of SC, we can identify some regions in some time period showing very consistent brain activities among the healthy control relative to the schizophrenia. So after a series of permutation tests, we found some brain regions as shown in panel A. The IC of the activity of these regions in the healthy control group was significantly higher than that in the patient group. At the same time, we can locate the time window in which this difference occurs. So as you can see on the plot C to E, this is an example. This means when watching this episode of the movie, the brain regions displayed on panel D has a significantly stronger inter-subject concordance or inter-subject correlation in the HC group than in the SC group. And panel E shows the time time step of when this happened. So you can see I put a right circle on the plot. This shows that the inter-subject correlation of the healthy subjects is pretty high when watching this content of the movie, but the inter-subject correlation of the schizophrenic patients are relatively low. So the difference is very significant. So by doing this, we can identify some regions, some brain regions, and we know that in some content of the under some content of the movie, these regions are of interest, and we may find some brain abnormality of the schizophrenic patients based on these locations at this time of the movie. So it's worth mentioning that in the primary visual cortex, as shown here, the purple spot, we also extracted the ISC of the brain activity within the two groups, and they behave very similarly over time, as shown on this panel. This suggests that the two groups are consistent in very basic visual information processing, and that this behavior of the two groups of subjects during data acquisition, such as paying attention to screen, are consistent. So now it is determined that healthy subjects have highly consistent temporal and spatial characteristics of the brain activity. We now proceed to construct a normative model. So panel A gives an example which reflects the activity curve of a brain area that has been identified in our previous slide. So you can see there's a fluctuation on the blue curve, and there is an arrow, and there are arrow bars indicating the variance among the healthy control populations. However, if you average the brain activity of the schizophrenic patients, the average curve appears very flat, meaning okay, there's no general trend among the patients. That means each patient is different from each other. So this is only an example. Actually, we have identified more than 2,000 regions or more than 2,000 voxels showing phenomena like this. So now we just use the blue curve as a template. We need to compare the brain activity curve of each subject, each individual subject with this template, and use the correlation coefficient to quantify their similarity. This enables individualized evaluation based on normative model. Of course, you can see, as you can see on panel B, if we integrate all the identified voxels, so the horizontal axis represents the index of the identified voxels, and the vertical axis indicates the individual subjects. But here I group them into two types according to the diagnosis. So the color shows the correlation between each individual's brain activity pattern, the similarity between each individual's brain activity pattern and the corresponding template. You can see for the normal control, on these voxels, their brain activities are very consistent. But for the schizophrenic patients, the consistencies are much lower. So this matrix on panel B provides a very nice feature for machine learning algorithms to learn. So if we train a support vector machine based on these features, we can actually easily identify schizophrenic patients. And actually, we can verify the performance of the classifier using independent data set. Panel C visualizes the relationships between healthy control and patients. Here you can see the blue dots and the red dots. As we predicted, we can see that the blue dots are closer to each other, indicating that they have a higher similarity, while the red dots representing the patients are farther apart. The classifier can accurately identify can correctly identify 19 out of 20 in the test data. So that means it guesses the subject's identity. Oh, it is correct very accurately. Okay, now on the panel D, you can see which regions are playing important roles for the support vector machine. So we also try to explain why the healthy control group has a higher consistency in the activity of certain brain regions. So we asked 20 college students to evaluate their feeling of the 18 video clips of interest we have identified from 9 aspects as shown on this slide, such as happy, angry, fearful. Is there emotional change? Is it related to yourself? Can you feel empathy, or does it require reasoning to understand the movie? So then we calculated the correlation coefficients between the ISC of each brain region in healthy groups and the scores of the various aspects. Here you can see the white dots and the gray dots indicate significant association, significant correlations. So we can find that some brain regions, the high inter-subject correlations in some brain regions are related to negative emotional scores, such as the right inferior parietal lobe. But for the other regions, there are some brain regions whose synchronized brain activity is related to the high score of reasoning. So this gives us an understanding of why people share similar brain responses when they see different contents of the movie. So the above study supports for the effectiveness of naturalistic imaging as an individualized brain function test. We next examined the reliability of the individual differences reflected in this paradigm. So this is a very methodological paper. I'm not going to walk through the very details. So briefly, in this study, we use the test-retest method to examine the reliability of the individual differences reflected by 2 types of natural stimuli, including natural viewing and emotional evoking movies. So in the natural side viewing, a video of natural sight without any human interactions were presented to the subjects. And in the emotional movie, short stories with different emotions are presented. So we recruited 24 healthy subjects and underwent 2 scans. The 2 scans were 1 week apart. So the subjects watched 2 videos in each scan session. So here we can get inter-subject correlation matrix for each session. And then we simply calculate the correlation coefficients between the 2 matrices to get a Pearson's correlation R, but in statistical term, it is also called stability coefficient. So the value of this stability coefficient evaluate how stable is the relationship retained between the repeated measures. So here you can see there are significant brain areas after very strict permutation test and corrected for multiple comparisons. So that means these brain regions, the signals in these brain regions can reliably represent individual differences. So they are mainly located in the temporal and occipital lobes and their injections. But particularly for the emotional evoking movies, we still can see some midline structure, and the parietal part of the brain shows high reliability. So in order to more accurately assess the reliability of the naturalistic imaging, we applied the same method to compare the reliability of some questionnaires and the neuro-imaging paradigm. In this figure, the color indicates the level of stability coefficient. Here in this bar plot, the color represents the same stability coefficient. We can see that for some regions in our brain, the stability actually were higher than the stability of some commonly used questionnaires, such as the state anxiety inventory. So these results support that the application of naturalistic imaging as a psychological test is possible. We also address whether the reliability of map we saw above relies heavily on the specific contents of the movie. In general, the reliability in some regions does not depend on the detailed content of the movie if the movie can evoke some emotions, especially the temporal parietal junction and superior temporal gyrus. Okay, the last study I want to present is an application of naturalistic imaging paradigm to evaluate the maturity of emotional functions in children and adolescents. So many studies have shown that adolescents exhibit remarkable individual differences in the structure and functional brain systems involved in social emotional processing. In adolescents of the same age, each individual may have a different process of neuron synapse pruning, but we still lack indicators to represent the functional maturity of an individual's social brain. So previous works have used self-report questionnaires such as emotion regulation strategy questionnaire and theory of mind task to reflect an individual's difference in social emotional ability. In this study, we developed a neuro-imaging approach for evaluating adolescents' maturity of brain functions in social emotional processing. So the first question we need to answer is, when processing social emotional information, does the similarity of adolescents' brain activities become higher with age? So according to the social environment of teenagers, we have edited a video containing emotional arousal, love, parent-child relationship, and school life. So we invited 40 students to rate the valence and arousal level on average 10 seconds of the clip. Here you can see there's fluctuation of the valence and the arousal levels along the movie. So we asked 111 adolescents aged between 9 and 17 to watch this video during brain functional scans. We applied the linear mixed model to test the association between intersubject correlation and age. As shown on the figure to the right, we found 16 regions of interest showing significant age effect. This addressed our previous question, meaning the consistency of brain activity patterns of adolescents can change with age. But what's the trend? Let's take a closer look at how the intersubject correlation of these brain regions change with age. So here we use 1.5 year as an age window to perform a sliding age window analysis. Here we show a majority of the ROIs show the trend of increasing intersubject correlation with age, and all ROIs intersubject correlation are relatively high and stable after the age of 16.5. So this means maybe we can use this age, relatively older adolescents, use as a reference group to evaluate adolescents of lower age. So in order to construct the normative model, we averaged the time series of the subjects older than 16.5 ages, 16.5 years, and consider the results as a brain activity template for each ROI. Then we defined a typicality index, which represents the similarity between an individual's brain response and the template brain response. For each subject, we can calculate the typicality for each ROI. We then evaluated the performance of the normative model to predict individuals' emotional regulation strategies and compare the performance of age and sex and the cognitive performance. On the plot to the left side, you can see when adding the brain response typicality can significantly increase the predictive power of the cognitive reappraisal, which is a critical component in emotional regulation in adolescents. Also, we have compared the effect of cognitive performance, such as the reaction time of in-back task. The results shows the same. We also validated the findings using an independent dataset. In this dataset, we show that including the brain response typicality can significantly increase the predictive power of children's performance in the theory of mind test. So this evidence shows that using how similar our brain respond to social emotional information with older adolescents or even adults, we have a brain-based indicator of the maturity of the social emotional functioning of the children and adolescents. So to support our finding, we further examine whether the typicality is driven by the emotional events of the stimuli. Here we derived dynamic typicality by using the sliding windows. That means in each clip of the movie, we are calculating individualized brain response typicality. And we employed an ANOVA model to including age and emotional ratings to calculate the effect of emotion content on the typicality index. So the results show that for most ROIs, the dynamic typicality either exhibited significant interaction effect between age and emotional ratings, or showed significant main effect of emotional ratings. So this result suggests that the individual differences in the brain response are really related to emotional contents of the movie. So that means we are actually measuring what the emotion evoke. Okay, so this is the conclusions based on the 3 studies. So combining naturalistic imaging and multivariate normative models is a promising paradigm for individualized, objective, and quantitative inference for integrated functions of the brain. And our research results support the reliability and effectiveness of this approach. Finally, I want to acknowledge all my collaborators and members in our lab. My collaborators for these studies include colleagues in Shanghai Mental Health Center and Beijing Normal University. And there are also funds from National Natural Science Foundation and Tianqiao Terrace Chen Institute. If you're interested in our work and the related works, you're welcome to visit our website, and feel free to write me an email if you're interested or have further questions. Thank you very much.
Thank you, Professor Young. Okay, Professor Young just introduced a new application of the neuro-imaging measures of the brain response in natural contexts. And he also introduced the reliability and the consistency of this method in reflecting individual differences, and the application of this method in adolescence. Let's see if there are any question for you. Okay. Okay, there's one question for you. Is the amount of brain activity that changes in depression more likely to affect physical behavior?
Yeah, actually, this question is more clinical. I'm not an expert on the clinical side, but I can try my best to answer it. Basically, our brain is interconnected. It's a network of neurons. So if something can change global brain activity, then definitely every aspect can be affected. So for depression, there are series of inhibitory control or higher level cognitive controls or emotion regulation strategies. And these functions are all related widespread neural networks. And for the physical behavior, they are related to more elementary functions in the brain, and usually we consider the top level integrated networks can have a top-down effect on elementary networks. So definitely, I think the more elementary networks, the more obvious that it's received top-down modulation. So my opinion is yes, definitely. The change in the brain activity in depression can affect physical behavior.
Okay, thank you. You can have a rest.
Right. Thank you very much.
Okay, next, we will introduce our last speaker, Dr. Jin Lu. She's from the first affiliated hospital for Kunming Medical University. She majors on social psychiatry and pediatric psychiatry, epidemiology for mental disorders in children and adolescents. Next, she will introduce her research based on a cross-sectional survey, which is about the prevalence of depressive disorders among the left-behind children and adolescents in a county of Yunnan province. Okay, it's time for Dr. Lu.
Thank you for your attention. Today I will give you a research of our study group. Our topic is the prevalence of depressive disorders of left-behind children in Yunnan province. It is a cross-sectional study. I come from the psychiatric department of the first affiliated hospital of Kunming Medical University. Our research is funded by National Natural Science Foundation of China. This study was approved by the Ethics Committee of Kunming Medical University. I will give you the four part of our research. First is the background. The left behind children in China is very often to be seen. Children and adolescents under age of 18, their parents worked abroad more than half a year. Both or one of the parents in the last year, their children for half a year these children will be designated as left-behind children. The particularity of LBC, the LBC have more mental health problems than other children, such as high risk of depression, suicide risk, and NSSI behavior. The current situation of LBC in China, there's some data during both life. The first is China Women's Foundation. In 2008, there were about 58 million LBC children in rural China. In 2030, it increased to 1 million. About 21.8% of the total number of our country. Our numbers of LBC is about 8 million in 2015. So we can see the number of left behind children increased from 2008 in 2015. The characteristic of depressive disorder in children and adolescents. Now, there are some particularly of this group. Firstly, there are higher prevalence of depressive symptoms in our country. It is reported as there are 44 to 55% who have depressive symptoms in Chinese children, and the total prevalence rate is 22.2%. And there are a high recurrence rate of depressive disorders in children and adolescents. Many parents asked us whether their children will be cured in the future. But the data give us that more than half of children who experience a different depressive disorder during adolescence experience at least one relapse during adulthood. And in this, in child and adolescents, there are high suicide rates. More than half of teen suicide victims suffer from depression, and making depression the most cause of suicide. And if children diagnosed as depressive disorder, there are very bad poor academic performance, mental retardation, suicide, substance abuse, and the conductive disorder. It's more harm than adult. We wanted to do some research about the left behind children in Yunnan province because there are little research in China. There are only 8 epidemiological surveys, cross-sectional surveys on the diagnosis of mental disorders in China, and there are only very few cross-sectional surveys of depressive disorders, not depressive symptoms, in children and adolescents. So we also found that there are no Yunnan province. Yunnan has a special geography location that economic background errors and of LBC. Our survey of disorders for children and adolescents is blank. And we did some papers. We searched for papers, found out that there are 3 national surveys of mental disorder in child and adolescents in the United States, and the most recent was in 2017. Turkey has conducted and published in 2019 about a national data. Our research site is located at Zhaotong area. Zhaotong area, in this picture, we can see yellow one part. It is located in remote areas of Yunnan province and lacks economy. It is the largest county for LBC in Yunnan province. In Yunnan province, every year, most of the parents go out to work in this county. So the number is increasing by year. And the current survey of the depressive disorders in LBC in Yunnan province. So we choose this county as our study site. To do this cross-sectional survey, we wanted to find out the prevalence and distribution of depressive disorders in LBC aged 10 to 18 in a country in Yunnan province. And we wanted also to analyze the correlates like social, psychological factors and other factors. And we wanted to try to explore the correlate factors of the MDD and depressive disorder. After we got more data, we wanted to provide a scientific basis for formulating strategies and measure for the prevention and control of depression among LBC remote areas in the Southwest. The part 2 is method. We including criteria in our study is that there's a self-administered questionnaire are using for some symptom screening.
您好，我是小一，请选择您咨询问题的类别。
for educational level, understanding the meaning of the questions. So only left behind the children over 10 years old and under 18 years old are included. The exclusion criteria including the guardian refuses to participate. The person refuses to
考试报名时间由各地人事考试机构自行设定，您可以通过以下提到获取信息：人事考试网网址。
Someone who have serious mental illness, serious physical illness, and age incompatibility, low intelligence, and school leave, dropout leave, language barrier, and so this is the flow chart of our study. This is a two-stage investigate. The first stage is the first step. We have as 500 students in our study. After that, we ask them to do some questionnaire and thus another questionnaire of PSAT screening, including PHQ-9 and JD7 and self-harm questionnaire. If one of them is positive, the students will include it in the second stage. The second stage is diagnostic interview. We use the Csds questionnaire to ask the children. After that, after the screening, we have 800 patients, 50 students included in our study, and the last number is 232 who have any diagnostic interview. The study was have finished from 2020 May 10 to June the 4th, 2021. It spanned 25 days. This is our sampling flow chart. We choose 21 one group. We choose 2 to 3 classes. We use the PPS method. We have 9 primary schools, 7 junior high school, and 5 high school. The last sample size is 5,252 sample. So this is our sampling flow chart. We also recruit some investigator. The
详见
investigator
中国人事考
includes
试网
2
首页
parts
考
of
生
the
问答第一个
students.
问题
The first
网上
one
报名
part
过
is
程
the
中
Kunming Medical University and the psychiatric department of the first affiliated university. The total investigator was 34 person, including 21 students and 13 doctors. The interviewer of Csds questionnaire are graduated students or doctors from psychiatric department. We use some surveys. The first is diagnostic criteria is DSM-5. This is the first survey in China we have founded that use DSM-4 criteria. The second, we use screening tool. The first screening tool is PHQ-9. The second is Csds. The last is adolescent self-harm questionnaire. The last one is Csds screening tool. So if one of them is positive, the students will recruit into the stage 2. The stage 2 we use diagnostic tools Csds PL version, which can give us the DSM-5 diagnosis of depressive disorder. We have a high quality control. The quality controller, including unified training, the project background related knowledge of introduction, onset investigation process and skills, questionnaire review rules, etc. And the interviewer of the Csds, including guaranteed professional background on unified training, including theoretical training on the use of Csds, diagnostic questionnaire, practitioner operational trials, and psychiatric ward training. Time is 1 week, and irregular online training sessions. And the other quality control, including the first stage of quality control, including on-site quality supervisors. The supervisor and the sport check the quality of coordination of all site questionnaires. The quality controller are in charge of each class and procedures. And the questionnaire interpretation method are required to unified. Both quality controllers review the questionnaire and to sign their names. In the second stage of quality control, when we make the face-to-face interview of Csds, the interview questions are conducted in a strict accordance with the interviewer manual. Regularly spot check the diagnosis accuracy and the consistency of the diagnostic, and diagnostic staff record voice to check. The statistic analysis of our data. Firstly, the data entry tool, we use Epidata or draw independent entry. The statistic tools we use are the R package survey package with the data and the design-based descriptive statistic and analyze. We also use design-based on univariate and multivariate logistic regression. Here comes to the part 3, the result. This is the social demographic characteristic of the total sample. The total sample is 5,252. After screening, the positive sample about 856. We can see the gender is similar in the total sample. The children in aged 13 to 15 years old are the most large sample. We can see most students are resident in rural areas, about 18%, and the grade is the large sample of the grade is junior high school students. There's some single child family, only 5.4% is single child family. This is a very small part. This is a very small comparison of single child, and the parental marriage is about 76% parent are married. The family economical level are most are stabilized because their parents are going out for work, and about 17% families are minimum guarantee. That's a poor economical level. We can also see this picture, see the sample distribution in this picture. So we can see the female-male sample are similar, and most of them are come from the rural area, and a lot of them are middle school students. Next, we will give you the lifetime prevalence of the depressive disorders. Depressive disorders include major depressive disorder, senior, and depressive disorder, NOS. The prevalence of lifetime MMPD is 2.62%, and the prevalence of dysemia is 1.1%, and the TDNOS is 0.82. The total percent of lifetime prevalence of depressive disorder in left-behind children in our study is 3.87%. And the table 2 is the 30-day prevalence of depressive disorder. The total prevalence is 3 to 3.45%. And for the major depressive disorder, the prevalence of 30-day is 1.92%. Next, we will give you the prevalence of depressive disorder by sociodomestic distribution. We can see both in lifetime and 30-day prevalence, female has a higher prevalence than male. In female, that's 5.72% prevalence of depressive disorder, and in age group, for age group, children aged 13 to 15 years old has a higher prevalence of lifetime and 30-day prevalence. So we give you others. In this picture, we can see clearly the prevalence of the distribution. For lifetime prevalence, we use blue color. We can see the most prevalence group is female, and the age group in 13 to 15, and for the resident area, we found out that urban left-behind children have more prevalence than rural LBC children. So and the single child has a higher prevalence of depressive disorder, and parent more. We can after that, we also analyze the correlates of the depressive disorder, also shown that female non-Han nationality middle school students have a higher prevalence than others, and students resident in rural area have less prevalence in their groups. This is the forest plot of major depressive disorder. We can also see that female students aged 13 to 15 years old and their family are in not married stage has a higher prevalence rate in the group, and the rural resident also protect them. This is the plot, forest plot of dysemia. We can see female junior high school and the parent is not married, and father's education, other education will influence the children's depressive disorder. And this chart shows us that the most of prevalence rate is the symptoms of the major depressive disorder. The most prevalence symptoms is depressive or irritable mood, and the second is depressive mood. The last prevalence is the sleep too much. This chart shows us some problem of sleeping problem. Most students of diagnostic as depressive disorder have a higher insomnia prevalence and lower sleep too much prevalence. The picture in the right shows us that there are three kinds of insomnia. The most prevalent rate is difficulty in falling asleep. The second is mid-segment insomnia, and the last is wake up early. So most students have the problem of difficulty in falling asleep. This picture gives us the conclusion about result about the incidence of NSSI and suicidal related behaviors in major depressive disorders. In our research, we can see the most prevalent rate is suicide. Just a limited time of thinking suicide, but in suicide ideation, there's a higher suicide ideation, about 53%. And about 11% attempt suicide. So in this area, there's a higher attempt suicide and the suicide rate in this area. So we, after this survey, we go to this county to do some CBT intervention for these students. This is also, this table shows us that the most prevalence symptoms of the depressive disorder by age groups for students aged 10 to 15 years old. The most prevalent symptom is depressive or irritable mood, and follow that is suicidal thought, insomnia, and worthless thought. So this can show us the symptoms of the depressive disorder. And we also conclude the symptom prevalence by gender. In female, the most prevalence symptom is also depressive irritable mood, and others are insomnia and NSSI males, but NSSI in females are lower than in males in our group. Here we come to the discussion part. Our prevalence is in the first column of the prevalence, Zhao Tong prevalence. So we can see our result is higher, the prevalence of depressive disorder is higher than other research in China and in the world. So we should pay more attention to the female groups and in the third age groups in 13 from 13 to 15 years old, and the parents' marriage also influence the patient, the students. They are in unmarried status. They are the students are easily get the diagnosis of MDD. Ethnic minorities students are have a greater risk than the Han nationality. Students who resident in urban have a higher risk of MDD. The age group is similar to the similar to the 30 to 50 years is at risk group. Conclusion. There were some limitation of our research. We only do some research for the left behind children. This is a cross-sectional investigation, the limited analysis of temporal and causal relationship between variables. The source information for this is come from the children themselves. So we will improve our study and for this study, we found out that there's a higher prevalence of depressive disorder in this area, and the prevalence is more. The prevalence rate is higher than others in our country and in the world. So we should pay more attention to the left-behind children in the southwest China or in Yunnan province and in this county. Thank you for listening. If you have...
Thank you, Dr. Lu. Okay, Professor Lu just introduced her research of the prevalence of depressive disorder among the left-behind children and the adolescents in Yunnan province. Her study found that there was a high prevalence of depressive disorder in this population, and the implication of this research is remind us to pay more attention to this population, and we should focus on intervention measures towards the left behind children. Okay, thank you. I think the time is limited.
Thank you.
Okay, today we have shared the new ideas and the progress about depression. And many thanks to the 4 speakers for their excellent presentations. And finally, thank you all for your participation. I think time is limited. It's a time to bring this forum to a close. And I wish to see you again 2 years later. Okay, thank you.